Prokaryotes is illustrated as such, allowing for multiple copies of RNA to be transcribed from one single sequence of DNA, which can be seen here, seeing multiple proteins from one DNA strand. We can identify key components of transcription, starting with the -35 and -10 region, which are promoter regions. This allows for the RNA polymerase to bind. The +1 region allows for the transcription to start. The RNA polymerase will want to come in and start transcribing the ribonucleotide triphosphate from the 5' to 3' end, but cannot do so without the sigma subunit. The sigma subunit combined with the RNA polymerase allows for it to bind to the promoter region, and together it is called the whole enzyme. The sigma subunit is illustrated as a person. Without the subunit, it won't be able to park the truck. Without the RNA polymerase, as illustrated by the truck, it won't be able to unpack and start transcribing that RNA. Once the RNA polymerase truck is parked, it will begin to transcribe, reading the DNA from the 3' end to the 5' end and making that RNA from that 5' end to the 3' end. You can see there's a termination site, but we'll get back to that at the end. We can label the strands of DNA, starting with the template strand. It will have its 5' of the DNA on the same side as the 3' of the RNA. There we can label the 5' and 3' end of RNA. We can also label the coding strand, and the coding strand is going to have the 3' of DNA and the 3' of RNA on the exact same side of each other. We can also label the direction of transcription. If RNA is being transcribed from the 5' to 3' end, based off of this illustration, transcription is going in the right direction. If the -10 region had accidentally been taken out or deleted from the DNA, this would deny RNA polymerase and its subunit to bind to that region, and transcription initiation is then lost. In the event that this is a gene that codes for an enzyme to metabolize sugar, the enzyme would not be transcribed, and therefore that person would not be able to metabolize sugar due to that RNA polymerase not being able to bind and thus transcription not being initiated. Going back out, we're going to talk about termination. Termination, it can happen in two ways: row independent and row dependent. Row independent termination is first presented with inverted repeats that form hairpin structures and are followed up by poly A and poly U template strands. AU bonds are relatively weak, and DNA and RNA can be pulled apart, ending termination. Row dependent occurs when the row binds to the RNA and moves towards the 3' end. The row molecule has helicase activity and thus relieves torsion and stress and unwinding of the sequence, causing transcription to end.